Good morning to all. So today we are discuss about in cell division mitosis. So already so studied in the previous class, the organisms is classified into two types, namely eukaryotic and prokaryotic, or unicellular and multicellular organism. The multicellular organisms is derived from the unicellular organism. The unicellular organisms undergo the cell division and to form a number of cells to produce a multicellular organism. So today, we are discussed in how to single cells is developed or multiply into multicellular by the process of mitosis. So first of all, cell. Cell are the structural and functional unit of all living organism. Cell is the structural and functional unit of the all living organism. The cell is classified into two types, unicellular and multicellular. Organisms is classified into two types. So unicellular and multicellular based on the number of cells found in the organism. Functions of cell. So nutrients of to energy. Each cells having its own sets of instructions. So nutrients to energy, and each cells having a own sets of instructions. Each and every cells having the own instructions, does not having any other external source of, does not depending of them any other organs or organelles or tissues like that. Cell cycle. The cell cycle is the event that makes a new cell by cell division through the sixth process, the cell cycle. The complete life cycles of the cell is called a cell cycle. So how do you read the previously in life cycles of butterfly, then life cycles of frog, then life cycles of some other insects, then plants, animals. So similarly, the cell cycle, the complete cyclic of the cell, or a single cell divided into two or more than two. The cell cycles having the six process, so they are first one interphase, second one mitosis, prophase, metaphase, anaphase, telophase, and cytokininin. So those are the six process involving in cell division of mitosis. So mitosis process having the six steps. So first one interphase, second one prophase, third one metaphase, fourth one anaphase, fifth one telophase, then sixth one cytokinesis. The end product or results of the mitotic cell division is single cell divided into two daughter. So first one mitosis. Mitosis is the mechanism that allows the nucleus of the cells to split and provide the each daughter cells with a complete sets of chromosomes during the cellular division. So mitosis means a single cell split into two daughters. The each daughters are having the same sets of chromosomes to the parent. So this coupled with the cytogenesis, divisions of the cytoplasm, so occurs in all multinuclear plants and animals to permit the growth of the organisms. This so coupled with the cytogenesis and karyogenesis followed by cytogenesis. So divisions of the cytoplasm is otherwise called as cytogenesis. Division of nucleus is called as karyogenesis. So occurs in all multicellular plants and animals to permit growth of the organism. So growth of organisms. First phase of the mitotic cell division, interphase. So during the interphase, cell replicates its nuclear DNA, so ensuring that when the cells divide at the end of mitotic phase, each of the newly so created daughter cells will contain a full sets of genes. During interphase, cell replicates its nuclear DNA. The interphase stages replicate the DNA or especially for the nuclear DNA. The DNA is found in the inside of the nucleus is called a nuclear DNA, outside of the nucleus of extra chromosomal DNA. Extra chromosomal DNA. The nuclear DNAs replicate during this process of interphase, so ensuring that when does it divides at the end of the mitotic phase. So each of these newly created daughter cells will contain a full sets of genes. A newly created or newly formed daughter cells having the full sets of chromosomes or genes. So it is the diagrams of the interface. Centrioles, there are two pairs of centrioles found in the cells. This is the plasma membranes or outermost living layers of the cells. Nuclear envelopes, it surrounded the nucleus. Then chromatids and nucleolus. The denser regions of the nucleus is called a nucleolus. It is the interface. This phase is occurs only the replicate of a nuclear DNA. Then next one, second phase, prophase. So during this first mitotic stage, it is the first mitotic stage, cell division stage. Mitotic stage, the nucleolus fats and chromatins, replicated DNA and associated proteins. So contains in chromosomes. During the first mitotic stage, it is the first mitotic stage for the cell divisions, the nucleolus fats and chromatin it's contains into chromosomes. The nucleus is fats. So nucleus is fats and the chromatin reticulums. The nucleus are having the four parts, namely nuclear matrix, the nucleus, chromatin reticulums, and nuclear envelopes. The chromatin reticulums is contained and to change into a chromosome. Change into a chromosomes. Each replicated chromosomes so comprised the two chromatids, both with the same genetic conformations. So each replicated chromosomes, the newly formed replicated chromosomes is composed of here two chromatids. So each chromosomes is consist of here two chromatids, both with the same genetic conformations. Both the chromatids having the same genetic conformations. I means homologous. Same characters are identical characters. The diagrams of the prophase so follows regions of the alveoles, then asteroids and prophase stages. The contains chromosomes having the centromere. The chromosomes contains of your two sister chromatids. Two sister chromatids. So see the picture. So it is the single chromosomes. These chromosomes having the four chromatids. I means four chromatids. The center regions located in the centromere. So both are having the sister chromatids and non-sister chromatids. It is the sister chromatids. The chromosomes is consist of two sister chromatids. Okay, so each chromosomes is consist of two sister chromatids. It occurs in the first phase of the mitotic cell division for phase. Next one, metaphase. So next, the nuclear envelopes break down and a large protein network is called the spindle. So attached to the each sister chromatids, the chromosomes are now aligned so perpendicular to the spindle in a process is called metaphase. So see that. So very carefully. The nuclear envelopes breakdowns. The outer productive layers of the nucleus is called the nuclear envelopes. The nuclear envelopes is breakdown on the large protein networks. Larger protein networks. Protein networks and the nuclear envelopes is breakdowns. A larger protein substance is formed is called a spindle. So the spindles is attached to the sister chromatids. The spindles is attached to the sister chromatids or the chromosomes. The chromosomes are now aligned. So perpendicular to the spindle. So perpendicular to the spindles. So spindles perpendicular structures வந்து நமக்கு chromosomes வந்து arrange ஆகுது. This process is called metaphase. So இந்த process நம்ம வந்து metaphase அப்படின்னு சொல்றோம். So it is the diagrammatic view of the metaphase. So see that it is the thread-like structures is nothing but it is the spindle fiber. So it's made up of only for protein substance. Only for protein substance. Metaphase plate. The chromosomes are arranged in the perpendicular to the metaphase plate. So this is the metaphase plate. The chromosomes are arranged in a middle portion of the cell. Chromosomes are arranged in the middle portions of the cells is called metaphase. Then next one, anaphase. First one, mitotic cell division first stage or first phase is prophase. Second phase metaphase. Then third one, anaphase. So I say that anaphase. So being as the centromere of each pair of chromatids split, effectively doubling the number of chromosomes. Cells or nucleus having the chromosome, each chromosomes having the single centromere. During the anaphase, the centromere of each pair of chromatids split. The centromere split into two. The centromere split into two. Each splitting centromeres having the chromatids, pair of chromatids. Each centromeres having the pair of chromatids, effectively doubling the number of chromosomes. So effectively doubling the number of chromosomes. The anaphase stage, the number of chromosomes is doubling. The number of chromosomes is doubling due to the splitting of the chromosomes. Once separated, the sister chromatids is now an independent. So once separated, after separations of the chromosomes, the sister chromatids is hence independent chromosomes, which its one centromeres. Which its one centromeres begins moving apart towards the opposite pole of the cells. So after separations of the centromeres, the each chromatids or sister chromatids is independent because of having the one centromere of the chromosomes. So after that, the centromere contain chromatids is moved to the opposite poles. The moment is made possible via two mechanisms. The moment is possible via two mechanisms. Them kinetochore fibers attached to the each centromere begins to the shortens, filling the chromosomes towards the folds. At the same time, the polar microtubules from the opposite ends of the cells form the cross bridge in the equatorial plates, so filling the poles apart the beginning longer. The movement of the centromere from center to the either poles occurs in the two mechanisms. The first one is contractions of the protein substance or kinetochore fibers. Okay, the kinetochore fibers is attached to the each centromeres. So being the short ends, contracles, pulling the chromosomes towards the fold, or otherwise, sometimes the microtubules, the tube-like structures, the minute tube-like structures is called microtubules. It's found in the polar regions or either pole of the cells. Is polar microtubules opposite ends to the cells form a cross bridge in the equatorial plate, sufficing the polar apart by becoming the longer. So see that it is the anaphase stages of the cell division. The chromosomes is splitting. The center equatorial regions arranged chromosomes. The centromere is split into two. So each centromere is having the sister chromatids. Those sister chromatids is independent. The sister chromatids is independence. So after that, the separations of the chromosome, the splitting chromosomes is move into either pole. The splitting chromosomes move into opposite poles by the contraction of the spindle fiber. The spindle fibers arises from the either fold of the cells and attached to the centromere of the chromosome. So after splitting of the chromosomes, the contractions of the spindle fiber, the chromosomes is moved to either pole or opposite poles in the anaphase. Next one, telophase. So the daughter chromosomes arrived at the spindle folds. Okay, the daughter chromosome, so that the slightly moving the chromosomes. So after completions of the anaphase, so completely move to the polar regions of the cells. The daughter chromosomes so arrives at the spindle folds. After complete the separations of the chromosome, the nuclear membranes begins to reform around the each group of chromosomes at the opposite end of the cell. So after completions of the separation, the chromosomes are separate into either poles. After that, the nuclear membranes begins to reform around each group of chromosomes at the opposite end of the cells. So now the chromosomes is moved to opposite poles on either poles of the cells. So after that completely separations of the nucleus or chromosomes so reappear of the nuclear membranes. The nuclear membranes is reappear reforms around the each groups of chromosomes. The nuclear membranes is forms the each groups of the chromosomes. So complete that new cells. When telophase is complete and a new cell membrane is begins formed. So after completions of the telophase or the karyogenesis, new cell membranes is begins formed. New cell membranes. Cell membranes means plasma membranes. Plasma membranes. So the final steps of the telophase involving the initiation of the plasma membranes cleavage between each of the new daughter cells to ultimately yield two separate cells during cytogenesis. So after that, the complete stage of the telophase, the initiation of the cleavage of cytoplasm or plasma membrane, the daughter cells ultimately yield two separate cells. So during cytogenesis, the next phase of cell divisions. So after completions of the telophase, the initiations of the cytoplasm or plasma membranes is cleavage to the cytoplasm into yielding of two daughter cells. Then telophase and cytogenesis is a simultaneous process. It is the nuclear region. It's also a nuclear regions of the single cell. The nucleus is the or center chromosome move to the opposite poles of the cell and reappears of the nuclear envelopes or nuclear membrane. A newly formed nuclear membranes, the single cell having the two nucleus. So each nucleus is covered by nuclear membrane. So after completions developed the nuclear membranes initiate the cytogenesis or cleavage the cytoplasm and plasma membrane. Cytoplasm and plasma membrane. Then last stage of the mitotic cell divisions is cytogenesis. The final stage in the process of cell divisions is known as cytogenesis. The end process of the mitotic cell divisions is called as cytogenesis, which usually begins during the later anaphase or earlier telophase. So before mitosis it start or initiate in them later anaphase stages and earlier telophase stages. Later anaphase and earlier telophase stages. So before the mitosis ends, before completions of the mitosis, the cytogenesis is initiate or occurs. As the nuclear envelopes is reforming and the chromosomes are decondensing. Okay, so after formations of the nuclear membrane in the cell, the chromosomes is split into two. The splitting chromosomes move to the opposite pole. So after completion, move the opposite pole of the chromosomes reappears or reforming of the nuclear membrane. The after formations of the nuclear membranes, the contents chromosomes is again changed into chromatin reticulums or decondensing or decondensing. During this stage, the cytoplasm is divided. During this stage, the cytoplasm is divided. Cytoplasm is divided. So these regions, cytoplasms is cleavage, the furrowing, the cleavage furrow is developed. The furtherly the cleavage furrows is developed into cleavage the cytoplasm. Cleavage the cytoplasms, the yielding or resultant of the mitosis to form two daughter cells in the mitotic parent. The parental cells to form two daughters. Two daughter cells. Okay, so today we are completed in the cell division mitosis. So mitotic cell divisions occurs in majorly for 5 times. So prophase, metaphase, anaphase, telophase and cytogenesis. So prophase. So overview for this today class. The prophase stage, the cells are nuclear membranes is disappear. The chromosomes is contains or chromatin reticulums is highly contains in the prophase stage. And the metaphase stage, the cells chromatids are chromosomes are arranged in the middle portions or mediated plate of the cells and attached to the formations of the spindle in the central or opposite pole of the cell. Then after that, anaphase, the newly formed spindle fibers is attached to the centromeres of the chromosomes and splitting of the centromere. So after the telophase, the splitting chromosomes is moved to opposite pole of the cells and reappears of the nuclear membrane. Then cytokinin initiate after completions of the karyogenesis, the initiation of the cytogenesis, the cleavage of the cytoplasms of the cells to form a two daughter. It is the stages involving in or process involving in the mitotic cell divisions. The significance or importance of the mitotic cell division. So it's maintain the number of chromosomes is same, or it maintain the same number of chromosomes to the parent. So major significance of the mitosis, it maintain the same number of chromosomes in the daughters or next progenies or next generation. So maintain the same number of chromosomes in the next generations does not lead to the variation. It maintain the heredity characters to the next generations. Not possible to form newly varieties or variations. These are the significance of the mitotic cell divisions.